Hey guys, hopefully you have already read section 10.2. If you have not, please stop this and go back and read that section. This is all going to make a lot more sense if you've gone over the material by reading it through one time. Alright, so section 1 was about genetics and Punnett squares and alleles, and this section is kind of, okay, so how do we make those gametes? Okay, so we said the gametes are the egg and the sperm. Well, where do they come from? So genes and alleles are passed from parent to offspring in the gametes. And the process of making a gamete is called meiosis. And it's a very unfortunately similar name to mitosis. I get that. But it is meiosis. And it involves, instead of in mitosis, we split the cell one time, and our cells that we created were exactly like the parent cell, right? In meiosis, there's a couple key differences. We're going to have two cell divisions. And so a cell splits in half, and we have two cells, and then each of those split in half. So we're going to have 4 genetically unique gametes. So they're each different from each other. Okay? And we'll talk about how that works out. But imagine if they were all the same, if your two parents had more than one child, they would be identical. And we know that that's not true. People could have lots of kids, and none of them are going to be exactly the same. Sometimes they look really, really similar, and we start to think, well, maybe they are, but they're not, I promise, unless they are identical twins. Okay, so remember, most cells in the organism have 2 copies of all genes, one from each parent. We had a word that we had that meant 2 copies of all the instructions, and that word was diploid. And again, you can say diploid, you can say diploid. It doesn't really make any difference. But our gametes, we don't want to have all the instructions, right? Because remember, they're going to get with another gamete to fertilize and make a zygote. So gametes have only half of the information, and so remember we had the word for them, which was haploid. All right, so when we think about diploid, we said we have one set of instructions from mom and one set from dad. So they kind of have 2 of everything, but they might not be exactly the same. Okay, they each maybe, if we're thinking about pea plants, so the instructions from mom and instructions of dad, they have instructions for height, they have instructions for seed color, they have instructions for is the pod smooth or constricted. But remember, instructions from mom and dad might be different. Like one gene might say tall from one parent and the other one would say short from the other parent. But we have chromosomes that have the same kinds of information, right? So we have some chromosomes that would have, information for traits ABC, and you would get one of those from mom and one of those from dad. And we call those homologous. And remember, this prefix is going to come up over and over in biology. Homologous, homo means same. So we have really similar chromosomes, one from mom and one from dad. So they have the same kinds of information in their genes, one from each parent. Okay, humans. We have 23 pair of homologous chromosomes. And we say pair because you get 23 from mom and 23 from dad, but they kind of match up, right? So we have one set of chromosomes from mom, and then each one of the chromosomes from mom has a homologous partner from dad. So we have 23 individual chromosomes from each parent. And so then they get together and they make the pair. If they are paired up, and this is true in most people, right? They pair up. You'd say we have 23 homologous pairs. Or pair, I think. Okay. Okay, so in meiosis, we have an important job. Okay, we need to make sure that in all of our gametes, all of our egg, and all of our sperm, we get exactly one of each homologous pair in each gamete. Okay, so this is a picture of all of the chromosomes in a normal cell. And so they're numbered 1 through 22, and then this must be a boy, because they have an X chromosome and a Y chromosome. But here's all the chromosomes. So for this particular individual, right, so there's chromosome 1. We'll say maybe the chromosomes on the left are from this person's dad, right? So this chromosome's from his dad, and this one's from his mom. And this one's from his dad, and this one's from his mom. So he would have 23 from dad and 23 from mom, and then they have their partner that they're really, really similar to. And if you look, they're not only similar in size, but if you look, they have those little stripes called bands. They have light spots and dark spots, and those spots are kind of in the same spots as well. Okay, let's keep going. All right, so again, meiosis is going to go through 2 divisions. Okay, and we call division 1 meiosis 1. And division 2, meiosis 2. They're very uncleverly named. The more science classes you take, the more you figure out how unclever scientists are when they name stuff. So meiosis 1 and meiosis 2 are both really, really, really similar to mitosis, which you are already an expert at, right? So we have PMAT, prophase, metaphase, anaphase, telophase, and we go through PMAT twice to get our 2 divisions. There's just a couple key differences between mitosis divisions and our meiosis divisions. But here's another similarity. Okay, before meiosis starts, again, just like in mitosis, DNA is copied. So we are going into meiosis with 2 copies of everything. Okay, and so here's a quick reminder. So if this was that chromosome from dad, when I copy it, I may attach itself to its copy just like this, right? So there's the original, and here is the copy. Okay, so I have a chromosome that has been copied, and it is held with its copy right there. And we remember that structure is called the centromere. Okay, so moving on. On the next page is a visual summary. Feel free to flip and look at that. I'll pull up those pictures as we're talking about each of those steps, but here we go. Big ideas. Okay, meiosis creates 4 unique gametes. Okay, whereas meiosis creates 2 identical copies. So in both the number of daughter cells that we are making and genetically how do they compare to each other, that is a key difference. Okay, in meiosis 1, we are going to reduce the number of chromosomes. Okay, so humans, we start with 46 total chromosomes, 23 pair, 23 from mom, 23 from dad. After the very first division of meiosis, we're going to split down from 46 chromosomes to 23. Okay, so that happens during meiosis 1. So right away we go from being diploid to haploid. So overall change in meiosis 1, for humans anyway, we go from 46 double chromosomes to 23 double chromosomes. And when I say double chromosomes, I mean that they are still attached to their copy. Okay, so let's look at what's happening here. Okay, so the top row of this picture out of your notes is meiosis 1. Okay, so I have all chromosomes. This particular one has 3 pair of chromosomes, or 6 chromosomes. Okay, so they're going to line up. Maybe blue is from this person's dad and red's from mom, right? So they're going to get together, go through PMAT. So they form, they line up, they get pulled apart, and now we have 3, 1, 2, 3, 4, 5, 6, all together, right? 3 here and 3 here. And we started with 1, 2, 3, 4, 5, 6. Okay, so right away, we split the number of chromosomes in half. But again, if you look, it's still attached to its copy. It still has that centromere holding it and its copy together. Okay, in meiosis II, okay, chromosomes are separated from their copy, very much like in mitosis, returning to single chromosomes. So we have 23 double chromosomes. Remember, double meaning they're still attached to their copy, down to 23, what I like to call single chromosomes. They're not with their copy anymore. So again, let's look at it kind of in stepwise here. So here's the 2 cells that formed after meiosis I. They each have their 3 double chromosomes. Okay, so they're going to line up down the middle in metaphase 2, get pulled apart. And if we look now in the cells that are forming, there are still going to be 3 chromosomes, but they don't have their copy anymore. Okay, they're single chromosomes. They have been separated from their copy. And let's maybe say here for single, that means not with its copy. Yeah, here's maybe a simpler way to kind of look at that. So we start with 4 single chromosomes. Okay, in interphase, remember this is before meiosis starts. In interphase, we're going to take each of these chromosomes and they're going to be duplicated. So now they are double chromosomes, they're with their copy. We're going to talk about the fact that they have different colors here in a little bit, or maybe in the next video actually. But the idea is now we have still 4 chromosomes. We had 4 chromosomes here. We have 4 chromosomes here, but now they're double chromosomes. They're with their copy. So we have 4 double chromosomes. Okay, so then this is going to go through meiosis I. And remember, meiosis I is when we reduce the number of chromosomes, but they're still with their copy. So now I have 2 double chromosomes. Okay, so I went from 4 to 2 in the first division. In meiosis II, we have 2 chromosomes here. After the division, I still have 2 chromosomes, but what's changed about them? Instead of being double chromosomes, now we have single chromosomes. So we have 2 single chromosomes. So again, first division of meiosis, we go from diploid, having 1 set from mom, 1 set from dad, to haploid. And then in meiosis II, we go from double chromosomes to single chromosomes. And I'm going to stop right there.